DNA is the blueprint for life. It tells organisms what to do. DNA is made up of 4 smaller bases that join together. DNA bases pair up with cytosine with guanine and adenine with thymine. The first step of creating a protein is transcription. The first step in transcription is RNA polymerase unwinds DNA. Next, RNA polymerase matches the bases with DNA. This makes an mRNA molecule. The last step in transcription is mRNA leaves and DNA winds back up. The finishing product of transcription is mRNA. The difference between RNA and DNA is that RNA uracil replaces thymine. Also, RNA only has 1 strand while DNA has 2. In RNA, the bases pair up with adenine with uracil, cytosine with guanine. The next step of creating a protein is translation. Translation is the process of building proteins from mRNA. The first step in translation is mRNA leaves the nucleus and attaches to the ribosome. Next, the ribosome will read the mRNA. It will begin once it reads the start codon. The start codon is methionine. Next, the tRNA will bring the amino acids to the mRNA. The protein is finished once the ribosome gets to the stop codon. The stop codons are highlighted in red. The finishing product of translation is a protein. Codons are made of 3 base letters that combine to make an amino acid. Amino acids are sort of like building blocks for proteins. Amino acids combine to make proteins. Proteins are molecules in our bodies that do most of the work in our bodies. Mutations can be caused by an error in DNA replication or damaged by environmental factors. Mutations can either be a good or bad result. There are 3 types of mutations: insertion, deletion, and substitution. Insertion is when an extra base gets added in. Deletion is when a base gets deleted. Substitution is when one base gets substituted for another. These 2 pictures are results of a good mutation and a bad mutation. Sickle cell is a bad type of mutation. It blocks up your blood flow. This is an example of some good mutations.